Today we will discuss how the vector pBr322 is used as a vector for gene cloning and how we can select the cells which contain the vector pBr322. So it is called as host cell screening. We go to the structure of pBr322. This is the first artificial cloning vector that was constructed by Francisco Bolivar and Raymond L. Rodriguez in 1977 in the University of California in San Francisco. Coming to the name of this vector, this P stands for plasmid, B stands for Bolivar and R stands for Rodriguez, the name of the constructors. This is a circular vector which is derived from E. coli and it is 4361 base pairs long. So you can see the circular DNA molecule. This is being used as a vector. So this is an artificial vector. Now coming to the number 322, this 322 distinguishes this plasmid from other vectors which were developed in the same laboratory like pBr325, pBr327, pBr328, etc. Now we come to the elements in the vector pBr322. It contains a region which is called as or replicon, which is responsible for the replication of the plasmid, because we are inserting our foreign gene in the vector and we want our foreign gene to get replicated along with the vector. So the vector should have the ability for replication. And this rep region confirms it is responsible for replication of this plasmid vector inside the host cell. Second, the ROP gene. The ROP gene encodes a protein which is called as the ROP protein, which will give stability to the vector plasmid inside the host cell, and it also controls the copy number of the plasmid inside the host cell, because plasmids can have many copies inside the same host cell. So that is being encoded by the copy number is being determined by the ROP gene. Both these sequences are taken from the plasmid PMB1. That is the original source plasmid from which these two sequences are taken for pBr322. Then there are two important genes which are the tetracycline resistance gene, in the presence of which the bacterial cell containing this plasmid will be resistant to tetracycline. So this is called as the tetracycline resistance gene, which will confirm resistance to tetracycline antibiotic. There is one more antibiotic resistance gene which is called as beta lactamase or BLA gene. It is the ampicillin resistance gene, because the presence of this gene will give the host cell the ability to grow even in the presence of ampicillin. That means it is resistant to ampicillin present in the medium. The host cells which do not contain the tetracycline resistance gene or the ampicillin resistance gene cannot grow in media containing tetracycline or ampicillin. So if these two genes are present, then the cells will grow in tetracycline as well as ampicillin in spite of the fact that they are antibiotics. This pBr322, as I already mentioned, is an artificial vector which is constructed from pBr318 and pBr320 E. coli source cell. Now the most important sequence of a vector is the ORI. ORI is the origin of replication, the sequence which determines the start of replication of any vector or any plasmid vector. And the ORI of pBr322 is adapted from the called E1 plasmid of E. coli bacterium. And the property of this ORI is that it multiplies faster than the host bacterial DNA. So you can have many copies inside one host cell and the replication is also faster. So along with the replication of the plasmid, our foreign DNA will also replicate. And the copy number of pBr322 is around 15 to 20 molecules in a cell. This pBr322 has got a number of restriction sites. Look at this vector. You can see a number of sites marked here. This is ECO R1, BAM H1, SCA1, PVU1, PST1, BSM1, PVU2, SNA1, ECO R5, Hind3, Cla1, etc. So, all these are restriction sites, which means that here the sequence present at this region in this vector, it is the palindromic sequence which is identified by the restriction enzyme EcoR1 for cutting. So, if EcoR1 enzyme is added to this vector, it will be cut at this region. Suppose we are adding the enzyme BAMH1 to this vector containing medium, the vector will be cut at this region. So, these regions are called as the restriction sites. So, this plasmid pBR 322 is having a number of sequences which are recognition sequences of different types of restriction enzymes. And this region is the ORI, which is determining the start of replication. And this region, this is the gene which confers ampicillin resistance. And this is the gene which confers tetracycline resistance. So, in the presence of the ampicillin resistance gene, any cell containing this plasmid will be able to grow in ampicillin containing medium. In the presence of this gene in the plasmid, any host cell containing this plasmid will be able to grow in tetracycline containing medium also. A host cell or a bacterial cell which is devoid of this tetracycline resistance gene cannot grow. Bacterial host cell which is devoid of ampicillin resistance gene cannot grow in ampicillin containing medium. So, the presence of these two genes confer resistance to the bacterial cells. Now, we go to how these two genes are being used for screening of host cells. The antibiotic resistance genes are used as markers in E. coli cells. Since normal E. coli cells are not resistant to any of the commonly used antibiotics. So, in an experiment, in a recombinant DNA experiment, we are linking a foreign gene to this vector, that is pBR 322. Usually, this vector is used to transfer foreign genes into E. coli host cells. These normal E. coli host cells do not contain the tetracycline resistance gene or the ampicillin resistance gene. So, they won't be able to grow in media containing these two antibiotics. But if the vector pBR 322 is present in this E. coli cell, then they will be able to grow in media containing these antibiotics. So, this property is used for checking whether this vector has entered into the host cell. That is what is called as screening. Now, we come to why we are using two antibiotic resistance genes as markers. We will just recollect the steps of recombinant DNA technology. In recombinant DNA technology, we cut a vector molecule and make it linear, a plasmid vector molecule to make it linear, and cutting is done using a particular restriction enzyme. It is customary that we cut the foreign gene also with the same restriction enzyme. Now, what we do? Now, we have to ligate the cut vector with the cut foreign gene to create the recombinant DNA, for which we are using the enzyme DNA ligase. But before that, this cut vector, since it is having sticky ends, it will have a tendency to undergo recircularization before insertion of the foreign DNA. So, we have to somehow prevent that. For that, we are adding the enzyme alkaline phosphatase. This alkaline phosphatase will cut out the phosphate groups from the 5' ends of the cut vector, so that it will prevent recircularization of the vector. Now, we will combine the alkaline phosphatase treated vector with the cut foreign DNA and join them using ligase. So, this is our experiment in which we expect the vector and the foreign DNA to join to form the recombinant DNA. But even though we add the enzyme alkaline phosphatase, there will be some vectors which will undergo recircularization, because the alkaline phosphatase cannot be considered as a 100% efficient enzyme. Before it can act, there will be many vectors which have undergone recircularization. So, that is one problem with this technique. Anyway, So that is the first step in which we cut the vector, treat it with alkaline phosphatase to prevent recircularization. We cut out the foreign DNA, mix the two and add ligase in the hope that the cut vector will join with the cut foreign DNA to form the recombinant DNA. But I told you there will be some vector molecules which undergo recircularization, but we cannot do the screening here. Now we proceed with the experiment. Now we will incubate this DNA solution which is containing our recombinant DNA, which may also contain the recircularized DNA, and we are incubating the solution with the host cells, and we use several techniques so that this DNA enters into host cell. So now we will come to our host cell. As I already told you, the host cells are normal E. coli cells which are not resistant to any of the commonly used antibiotics. So such host cells are pre-treated and then they are mixed with this DNA solution. So after the experiment, we will have 3 types of cells remaining. After the transformation experiment, there will be 3 types of cells remaining. They can be called as non-transformants, transformants but non-recombinants, and recombinants. So what are they? First one, we go to the non-transformants. After all this treatment, in spite of all this treatment, there will be some E. coli cells which have not taken up any DNA from outside. So they will remain as normal E. coli cells. They are called non-transformants. They are the host cells without vector. That means in spite of following all these steps in recombinant DNA technology, there will be some E. coli cells which didn't take up any DNA from outside. They are called as non-transformants. Now we are having transformants. There will be E. coli cells who have taken up the vector, the plasmid from the solution. They are called as transformants. So those E. coli cells who have taken up the pBR 322 vector from the solution into their cytoplasm can be called as transformants. But now, there are 2 types of transformants. There will be some host cells which have taken up this pBR 322 vector from the solution, but they have taken up a recircularized vector which is not containing any foreign DNA. Such cells are said to be transformants, but they are non-recombinants because they are host cells with vector, but they do not contain the inserted foreign DNA. They contain the recircularized vector. We don't have any use with such cells. Now we go to the third category. They are transformants which have taken up pBR 322, and this pBR 322 is containing the foreign gene inserted. Such cells are said to be recombinants. So they are host cells with recombinant vector DNA. So the 3 types of host cells which we have to expect after a procedure are the non-transformed cells which have not taken up any vector from outside. They are the normal E. coli cells, the transformants but non-recombinants who have taken up the recircularized vector which is not containing the foreign DNA, and the recombinants which have taken up the vector with the foreign DNA. We have to screen these cells to finally recover only the recombinants because we want only the recombinants. So how can we screen them? Screening is done with the help of antibiotic resistance genes. So for that we will go to the structure of this pBR 322 again, where we can see this is the tetracycline resistance gene. This is a sequence of this gene. Within the reading frame of this gene, you can see many restriction enzyme cutting sites like NHE1, BAMH1, SPH1, SAL1, etc. Suppose your foreign DNA was cut with the enzyme BAMH1. We usually follow the custom that if you cut the foreign DNA with a particular enzyme, you use the same enzyme for cutting the vector also. So here you will cut the vector using the enzyme BAMH1. So what happens? The vector is cut at this region. Now you mix it. After alkaline phosphatase treatment, you mix it with the foreign DNA. Since both are cut with BAM H1, they will be having sticky ends, so they will be joining with each other with the help of the enzyme ligase. So what happens? The foreign DNA will get inserted at this region. But insertion of the foreign DNA will alter the sequence of this tetracycline resistance gene, because the foreign DNA has got inserted into the reading frame of the gene, that is tetracycline resistance gene. So if foreign DNA gets incorporated here, the property will be lost. That is, the property of tetracycline resistance will be lost. This is called as insertional inactivation. So if you are using BAM H1 for cutting pBR 322, what happens is that the cut will be occurring at the tetracycline resistance gene. And if a foreign DNA gets inserted in that region, this vector will no longer be able to confer tetracycline on the host cell, because its reading frame is disrupted by the insertion of the foreign DNA. That is why this technique is called as insertional inactivation. So now we will go to the screening. As I already told you, if the foreign gene is inserted within the tetracycline resistance gene, the plasmid vector will lose tetracycline resistance, but the ampicillin resistance property is intact. Now we will just go to the three types of cells which we are expecting in a medium: the non-transformants, the non-recombinant transformants, and the recombinants which we want to proceed with. The non-transformants are normal E. coli cells which didn't take up any vector. So they don't have ampicillin resistance gene or tetracycline resistance gene. So they cannot grow in ampicillin or tetracycline. Second, the transformants which have taken up this vector, but the recircularized vector without the foreign DNA. So they will be having intact ampicillin resistance gene as well as tetracycline resistance gene. So such cells will grow in ampicillin and tetracycline containing medium. Now we go to the third category, the recombinant cells. The recombinant cells are those cells who have taken up this plasmid which is containing foreign DNA inserted in the tetracycline resistance gene site. So what happens? It will be having this ampicillin resistance gene intact, but the tetracycline resistance property will be lost because of the insertion of the foreign DNA. So how will the cells behave to these two antibiotics? The recombinant cells will grow in ampicillin because this gene is intact, but they do not grow in tetracycline. So we can see three different responses of these three types of cells towards the two types of antibiotics. That is, the non-transformants do not grow in ampicillin or tetracycline. The non-recombinant transformants can grow both in ampicillin and tetracycline. Whereas the recombinants, they will grow in ampicillin but not in tetracycline if the foreign gene is inserted within the tetracycline resistance gene area. So how can we screen the host cells for recombinants? So after the transformation, you will have to plate the cells into an ampicillin containing medium and you have to incubate them until the colonies appear. So you are taking these cells and allowing them to form colonies in an ampicillin containing agar medium. Which cells will grow in the presence of ampicillin? We will just check. The non-transformants do not grow in ampicillin, whereas the non-recombinant as well as the recombinant transformants will grow in ampicillin. So in the first step, when you plate the cells into ampicillin containing medium, you will see some colonies appearing. All these colonies are transformants. The non-transformed cells are ampicillin sensitive, so they do not produce colonies on the selected medium. Now we come to the colonies. Among these colonies, there will be some formed from the non-recombinant cells and there will be some formed from the recombinant cells. You only want the colonies formed from the recombinant cells. And how is their reaction to the next antibiotic tetracycline? The non-recombinants grow in tetracycline, whereas the recombinants do not grow in tetracycline. So your next task is to find out these colonies which further will not grow in tetracycline. So only a few transformants contain recombinant pBr 322 molecules, others will be containing the recircularized plasmid. So to identify the recombinants, these colonies are to be replica plated onto another agar medium that contains tetracycline. So after incubation, what you can see? Some colonies will regrow but others do not, and they are the recombinants because they are the ones which have grown in ampicillin containing medium but which do not grow in tetracycline containing medium. Those that do grow consist of cells that carry the recircularized vector, and those that do not grow on tetracycline are recombinants. They are resistant to ampicillin but they are sensitive to tetracycline. So you can refer back to the original ampicillin agar plate and you can reveal the positions of these colonies, and this enables the samples to be recovered for further study. So we will see how. Here you have a master plate where you can see different types of colonies formed from different types of cells. So you can consider this as the master plate, which will contain colonies from non-transformed cells, colonies from non-recombinant cells and colonies from recombinant cells. So you have to screen them. So this is your master plate containing all the three types of colonies. So what you have to do is you have to make a replica plate. How is it done? You are covering a wooden plate with a sterile velvet and you are just apposing this to this plate. So from all colonies, some of the cells will enter into this sterile velvet cloth. Now using this velvet cloth, you will develop new colonies in a new agar plate which is containing ampicillin. That is what is called as replica plating. That means you are making exact copies of these colonies onto other agar plates which contain ampicillin, tetracycline, etc. So that is what is called as replica plating. So here you have a master plate, master agar plate which is containing the three types of colonies. You are replica plating it onto an agar plate which is containing ampicillin. Now in the presence of ampicillin, you can see only some of these colonies prop up. The original master plate had this much number of colonies. We will just count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. But here, out of these 12 colonies, in the ampicillin containing medium, you can only detect 6. That means the other 6 are non-transformants which have not taken up any plasmid from outside. They do not contain the ampicillin resistance gene. That is why they cannot grow in ampicillin. So the first step is over. You have found out all those colonies which contain the pBR 322 vector because they all contain ampicillin resistance gene. Now you have to select those cells in which the tetracycline gene is inactivated because of the presence of the foreign gene. So you just replica plate this onto a tetracycline containing plate. You can see the 6 colonies here, but once you replica plated it to an agar plate containing tetracycline, only 3 colonies appear. So I can say 1, 2, 3. These 3 are colonies formed from cells which have the ability to grow both in ampicillin and tetracycline. They are the non-recombinant cells which have taken up this vector in the recircularized form. That is why they grow in both ampicillin and tetracycline. What type of cells do you want? You want those cells which grow in ampicillin but do not grow in tetracycline due to the presence of the inserted foreign gene. So here, these are the colonies growing in ampicillin. Out of them, these are the colonies growing in tetracycline. But whom do you want? You want those colonies which grow in ampicillin but do not grow in tetracycline. They are 1, 2 and 3. They grow in ampicillin but do not grow in tetracycline. The blue ones grow both in ampicillin and tetracycline. They are non-recombinants because they contain the recircularized plasmid. We don't want them. We want those cells which contain the plasmid with the inserted foreign gene, which will be growing in ampicillin but who will not be growing in tetracycline. They are these 3. 1, 2, 3. So that is how you use the technique of insertional inactivation to screen the bacterial cells which are recombinants. Now you will proceed with your experiment only with these recombinant cells. You do not want the non-transformants or you do not want the non-recombinants. You only want the recombinants. So that is how screening is done.